This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. From Microbe TV, this is TWiM, This Week in Microbiology, a podcast that explores unseen life on Earth. Hi, everybody. I'm Vincent Racaniello, and today is March 3rd, 2016, and this is episode 123. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there.
Welcome back.
Thank you.
I hope you have recovered.
I did. I had a nasty virus.
Well, they can do that. Viruses are sometimes nasty, but I like to tell people they're mostly good.
True.
But not everyone believes that, that's the problem. Also,
well, they didn't alter my evolutionary
trajectory.
Orbit, but maybe they did.
Well, you missed the TWiM. I think that's a significant alteration of evolutionary trajectory. Also joining us today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
And how are you? Are you in warm weather yet?
Oh, it's gorgeous here. The sun's been out all week. It's been about 75 degrees, and everybody's talking about going to play golf. So it's unfortunate Michelle's not going to join us today because I could tell her about the greens are looking good, and not that I play golf, but...
Here it's freezing, Michael.
I'm certain.
Yeah, I woke up this morning, it was -4 Celsius, and we're still in winter. Yeah, it's all cloudy today. I have a window behind me. On the last episode of TWiM 122, we mentioned the Tardigrade T-Shirt. Live tiny, die never. Tardigrade. Nice little picture of a tardigrade on it. Anyway, if you want to buy that T-shirt, it's at one of these sites where enough people have to sign up first before they'll make them. So here's the link for that if you're interested. This has nothing to do with TWiM or ASM or me or anybody that I know. We don't make money on it. It's just a cool T-shirt. And the link is bit.ly/TARDIGRADET. So that's bit.ly/tardigrade with a T appended to that last word. It's a really nice shirt. Check it out. Today we have a special episode for you. We have a guest, which we don't often have here on TWiM. And he is a colleague of mine here at Columbia University Medical Center. He is in the Department of Systems Biology, which is a relatively new department here, and he's a professor there. His name is Harris Wang. Welcome to TWiM.
Great to be here. Thanks so much.
Now, you've been here for 3 years, you said?
Yeah, 3 years and 2 days, actually, yeah.
You used to be on my floor, like 20 yards away, and you used to come by and chat all the time. And now you've moved to a better lab, and we miss you here. So I thought I would get you back and you could tell us about what's up in your lab.
Yeah, it's great to be here. Yeah, it's, just even having to walk across the campus in the cold winter, it really made me miss this place even more. So
it's too bad that distances are impediments on universities. People don't want to walk across the street, and it really impairs collaboration. So let's talk a little bit first about your history. Before you came here, you were with George Church, is that right?
Yeah, that's right. I did my PhD with George Church at Harvard Medical School in the Department of Genetics. And then I've kind of worked on a number of genome engineering, genome editing technologies while I was there. And then
came here.
So
yeah. You came here without a postdoc.
Well, so I spent about 1.5 years, 2 years at the Wyss Institute for Biologically Inspired Engineering, which is this multi-interdisciplinary institute that George Church actually is involved in, but it's headed by Don Ingberg at the Harvard Medical School. And it's basically a combination of people trying to make tissue engineering, organ-on-chip systems, to robo-bees, to DNA origami, to genome engineering.
Wow.
And so everything you can think about, kind of integrated all into one place. And I was a Wyss Technology Development Fellow there for about a year.
And you're independent, so it's sort of like a postdoc.
Right? So it was kind of it was like a postdoc. I was there for a year, and then I received one of these NIH Early Independence Award from the Director's Program, and then so that basically gave me an opportunity to start a lab while I was still
Nice.
at Harvard, and then I kind of jumped.
Before you
here.
went to Harvard for your PhD, where'd you go to college?
I was at MIT for undergrad, so I had
This guy's too smart.
I stayed in Boston for a really long time, and my whole plan, up to that point in terms of my life goal was that I was going to move every 9 years, so I'd never say I'd stayed in any place for more than a decade. And so I had actually moved to this country when I was 9. From China, and then and I moved to Boston for college again, and so I figured I would do a 4-year undergraduate degree, and then I would finish in 5 years for my PhD, and then I would just go off to somewhere else. So.
Where did you grow up in the U.S.?
I was in Salt Lake City, Utah. Actually,
So you know how to ski.
Well, that was one thing that I guess everyone there is supposed to know how to ski, but I never really kind of took on those endeavors too much. I guess being from a family of a single child, they were like, we don't want you to risk
you get hurt.
Yeah.
So you had the helicopter parents making certain that you didn't do hang gliding and other risk-taking behaviors.
Yeah, so those were all done, in college and later on. So,
So I have one of my podcasting colleagues is at University Salt Lake.
University of Utah. Yeah.
Nels Eldey, do you know that name?
Name's familiar.
Evolutionary biologist. So we do a podcast called This Week in Evolution, TWiVO, and we just started it. And Nels, we do all kinds of things. We do viruses, bacteria, butterflies, yeast, anything that evolves, we do. And what you can do now with evolution is amazing. So had you always wanted to be a scientist were your parents scientists?
Yeah, so my dad was a scientist, and so he studied mechanical engineering, applied mechanics, and so kind of biomedical engineering. And I always thought I would do something related to that kind of biomedical engineering, something where you could be quantitative in the biological sciences arena. Which is why I studied math and physics when I was at MIT, and so it was even though I was definitely planning on doing something in biology, but then my kind of interest really kind of gravitated towards more molecular level.
So I've never met George Church, but from what I can see from a distance, he seems like a really smart guy. Is that right?
Oh yeah, I mean that's I think that's very much true, and I think he's one of the smartest persons I've ever met. I think he also has this inspiration about him and the way he approaches his work, which has really helped me in terms of thinking about my own work that really is grounded on thinking about the future and really positioning your work in the context of what is going to be interesting and important 5, 10, 15 years down the line. And so and that's also seen in the types of work that he engages in, from kind of genome editing to synthetic biology to DNA sequencing.
It's a real big picture of things. I mean, he doesn't want to look at one gene and one bacterium. He wants to look at all of them. Yeah. Right?
Yeah.
Have you brought that philosophy with you here to your lab?
Absolutely. I think that's something that's very contagious. I mean, this idea of multiplexing, right? That instead of studying one thing at a time, you would be able to do that across many different things, but you could study it simultaneously. And that's really what has revolutionized things like next generation sequencing, is that instead of sequencing individual molecules, you're essentially sequencing the entire genome all at the same time.
I was trained, and probably I don't want to speak for Elio and Michael, but I bet they were the same way, to look at one gene and just study it your entire career, or just a few genes. I worked, for example, on one virus, polio. We got the sequence, and in fact, I was at MIT for a couple of years. It took me 1 year to get the sequence of the polio genome, and then I studied just that for the next 15 years. But now, if you want, you can get thousands of polio sequences from all over the world, which we couldn't do.
Yeah, that's right. I mean, and the technology changes so much, right, in such a short period of time. And so some of the things that we might never have thought to be possible are really kind of at the our fingertips now. Sure. And so it really kind of takes some level of appreciation for how technology could transform our ability to investigate natural processes and kind of the things.
Well, you got to remember, when I was a postdoc, that was the beginning of recombinant DNA. That was pretty revolutionary. And then there was DNA sequencing, Maxim and Gilbert. Didn't George work for one of those?
Yeah,
guys?
he was a graduate student with Wally Gilbert.
Wally Gilbert.
Yeah, that's right.
And then it just keeps going, PCR, DNA microarrays, deep sequencing, and more proteomics.
There's also another change that happened going away from a single gene.
Yeah.
It's going away from a single organism.
Right.
Oh, yeah.
We all were used to having a few model systems, D4, Lambda, a few animal viruses, and E. coli.
That's right.
That was it. I mean, there were a few other things in the world, but the view that we have today, which I think in good part is due to metagenomics, is that there's an infinitely large universe there for us to study, and it's all fascinating.
Yep.
Very different outlook.
So what brought me to asking you to join us today, Harris? I got an email from one of our listeners. I think it was Dennis, and he said, could you guys just explain MAGE? Maybe just take 15 minutes. And I said, no, we're not going to explain it. We're going to go right to the guy who invented it. And so I pulled up a couple of your papers, and one of them is this Nature paper, which in scientific terms is quite old now, 2009. And maybe you're going to tell me you don't even do this anymore, but I thought we would talk about it a little. It's called Programming Cells by Multiplex Genome Engineering and Accelerated Evolution, which is MAGE, right?
Yes.
Are you a gamer by any chance?
I used to play more games now than now, but
I used to be. So MAGE is in games, right?
Yeah. That's
And now he just writes grants.
That's right. Some form of that.
Now you're the first author on this paper, and it's out of George Church's lab. So I presume this is a big part of your work.
Yes.
Right. This is a way to look at many genes in many organisms, right? Did you guys come up with this together, or was it your idea? How did that work?
Yeah. So when I first kind of joined the lab as maybe a clueless first year graduate student, there was a kind of a emerging field that was coming to be formed in the realm of synthetic biology. And this is the idea that maybe we could, instead of doing the recombinant technologies, we would be able to actually develop more quantitative and technology-driven means to engineer microbial systems. And if we could do that, we could potentially revolutionize many different areas. So microbial engineering for applications, things like metabolic engineering, for developing new therapeutics that are microbial-based. And so that really drew me to kind of this field. And so one of the key challenges that the field had faced was that at that point, you couldn't really do genome editing or genome engineering very well. And it was mostly based on homologous recombination using double-stranded DNA. You would have to introduce some type of antibiotic selection marker where you require a crossover into the genome, and you select for the antibiotic marker, and that allowed you to do a genetic knockout, right? And that really took a few days to be able to do if you had a high-efficiency homologous recombination system. But that was to the extent for which we can actually do site-specific perturbation or modification of the microbial genome. And so I thought that was actually a very great challenge. And if we could somehow develop a next-generation set of tools to be able to allow us to do single nucleotide kind of editing of the genome, that would be quite transformative in terms of thinking about what types of things that we could do to engineer the microbe with new functionality.
So you want to be able to, instead of knocking in sequences on plasmids, you want to be able to edit right in situ on the genome.
Exactly.
Okay.
Right. And so, and part of the origin actually of this technology, and I think this is in general true for any technology that is going to be utilized on the long term, is that we had a different goal in mind, which was that we had a project where it was called Recoding the E. coli Genome, where we're trying to convert one of the synonymous stop codons in E. coli to its cognate, the TAG stop codon to the TAA stop codon. And the idea was that you would then free up the stop codon to allow incorporation of a non-natural amino acid. Right. And so expansion of the genetic code, if you will.
And we actually did that paper on TWiM.
Yeah.
On TWiM. Michael Schmidt, that was yours, right?
That was with me.
Right.
I had the honor of presenting that in
A couple of years ago, right?
Yeah, it was a while ago. It was in the early days of TWiM.
Yeah. Yep.
Yeah. So that's very interesting because, this paper actually came out of that for the need for introducing, we were trying to introduce something like 321 single nucleotide changes to the bacterial genome at very specific locations. And there was just no technology to try to do that. And so we looked at all the different ways that we could try to attempt to do this. And we found one such methodology that had actually been described by Don Court at NCI. Also, Francis Stewart had something very similar, where the idea was that you could introduce small pieces of oligonucleotides, single-stranded DNA, by electroporation or some type of DNA transformation method, and when you also overexpress or express a phage single-stranded binding protein, in this case the lambda protein, the beta lambda protein, you could then allow these single-stranded oligonucleotides to get incorporated into the chromosome at very, very high efficiency, something like 30 to 50% of the time at every replication stage. And the mechanistic of how that works is really based on what we think is like an Okazaki fragment-like process. Doing replication, the lagging strand is essentially incorporating these short oligonucleotides. And so by artificially introducing these oligonucleotides with specific homologies to the genome for which you're trying to edit or perturb, you could get them to somehow site-specifically incorporate.
Yeah. So it's dependent on DNA replication, right?
Exactly. And so you would have to have the cells actively replicating, and the limit of actually why, we can get 30 to 50% is actually because in doing semi-conservative replication, half of the chromosome is replicating in the negative strand. And so the oligos are essentially incorporating in as a complementary sequence. And so that was kind of the origin of what we thought could be a technology that allow us to do single-stranded, a single nucleotide incorporation into the genome at very high efficiency. And so, being in George Church's lab, we decided to multiplex this protocol. And so we said, instead of just doing one oligo at a time, what if we just introduce pools of oligos that could target one or multiple positions across the genome? And we actually found that there are mechanism for which there's a lot of synergy, such that incorporation of a single nucleotide, single oligo, also oftentimes allowed incorporation of multiple oligos.
Is there a distance limitation for that?
There is a distance limitation, and actually, if you could select for the incorporation at the beginning of the replication fork, then you can actually get very high efficiency incorporation of all the positions behind that replication fork.
Wow.
But how do you know where the replication fork is going to be?
Right. So we don't, and because the cells are asynchronous in terms of their cell division, there's a distribution of where the replication forks are. And so some of them are going to be in front of the position that you're trying to target, and some of them are going to be behind. And obviously those that are going to be behind are no longer kind of available for this oligo incorporation. But this process stays on for multiple generations. And so even if you don't catch it in the first replication, you can get the second or tertiary replication.
So your technique is dependent upon the asynchrony of bacterial growth cycles. You can never really get a synchronous culture. And that's the magic in the system, is that they're all at different phases of growth when they're replicating. And so you can actually select the global population simply because your community is at different various stages.
Yeah, but you can synchronize cultures, and you may gain something from it, because then you can do it. You can target the portion of the chromosome you want.
No, it's only when you're in exponential growth, the cells.
Right.
It's a population.
You can get a bacterial culture to divide in synchrony and to replicate in synchrony.
But the individual cell is not. I had this.
Sure.
No, we can bring Arthur Koch on, and he'll torture us all with. You can never actually precisely get them all to be in the same spot at the same time. It looks like it on a curve, but the individual is not.
Well, the other thing is also that oftentimes there are multiple replication forks that are occurring simultaneously within the cell. And so, it actually takes the bacteria more than 40 minutes to double in terms of making all of its DNA. But the doubling time actually is, 20 minutes or so, a little bit over. So it actually is making more copies of itself simultaneously. So within each, at least in exponentially growing E. coli cell in rich media, there's probably 4 to 8 copies of the chromosome that are at various stages of replication. So that also introduces an additional layer of a complexity in terms of synchronization. But yeah, we've tried some aspects of trying to synchronize the cells, and generally the efficiencies are not as high as what we observe. But that's definitely something that is worth exploring.
So in practice, how many genes would you target at once? You can do 2, 4, 10, 100, or there's no limit? Yeah,
for generally what we do in the laboratory today, we probably do 20 or so at a time. It also, it's kind of dependent on what you're interested in doing, right? And so you can this technology allows you to do site-specific mutagenesis. And if you're really interested in a particular genotype, then you need to have a corresponding assay to be able to identify those mutants. Right. But if you're not interested in a particular genotype, but rather a phenotype of some sort, such as being the ability to grow in a solvent a little bit better, then you might not care so much about the specific genotype as versus purely finding the variants that work. Right. And so if you're looking for the genotyping, that's obviously a challenge because now you have to be able to measure 20 different positions across the chromosome in many, many different cells. Yeah. And so what we came up with as part of this MAGE protocol was using allele-specific PCR that's multiplex. So it's a multiplex allele-specific PCR where in one PCR reaction, we're amplifying, we're querying the presence or absence of a particular mutation, but we do that 10, 20 positions at a time. Oh, yeah. And so that helps with this. But that's always a challenge.
So you would...
I don't know if this is the time to ask the question, but how has CRISPR influenced your work?
Yeah. So, I think CRISPR is a really fantastic technology, and I think it's really transformed the mammalian genome engineering, genome editing field. I think in terms of bacterial genome editing, it's still somewhat limited because of this issue of homologous recombination, right? So in mammalian cells, you rely on endogenous homologous recombination to then fix the double-stranded break that's generated by the CRISPR-Cas9 system. In bacteria, in general, there's not really good homologous recombination systems that is able to fix itself, usually because it's usually not a diploid genome, and so it doesn't even have another copy of itself to fix in some way. And so, which is why it's so successful as a immune mechanism in bacterial cells. It's basically able to chew up DNA. And so, from a bacterial genome engineering perspective, it's very useful as a negative selection as opposed to a positive selection, which is, you have a sequence that you're trying to get rid of. Maybe it's a wild type sequence. Let's say you were trying to engineer a mutation into the chromosome, and you can only get that mutation at a very low frequency, 10 to the minus 4, 10 to the minus 5, not minus 6. You're not going to be able to identify that mutation easily. But if you had a CRISPR system that targeted the wild type sequence, then you can suddenly increase the rate at which those mutant variants are found in the population. And so that's been where it's been very useful in the context of bacterial genome.
Yeah, makes perfect sense. That's great. This episode of TWiM is sponsored by Microbe Magazine Podcast. This is a production of the American Society for Microbiology that runs together with Microbe Magazine. It's a monthly show, and it is hosted by Jeff Fox, who is the current topics and features editor at Microbe Magazine. If you are an ASM member, you may know you get this magazine in the mail every month. It's also online, and each podcast episode coincides with an article in the current issue. For example, episode 8 of Microbe Magazine Podcast focuses on producing yeast strains so that you can make lager beers with new flavor notes. Pretty cool stuff, so you should check it out. You can find Microbe Magazine podcast on iTunes. Just search for Microbe Magazine or go to microbeworld.org/MMP. So in practice, you have pools of oligos and you introduce them into cells by electroporation, right?
Yeah.
You have to do this multiple rounds. I think in this paper you have an apparatus that you made so it can automatically happen. How does that work?
Yeah, so the protocol is basically you introduce oligos into the cells. You grow the cells into exponential phase, and then you induce the single-stranded binding protein beta to be expressed, and then you transform.
So that has to be on a plasmid in your cell of interest. Is that
it's
right?
either on a plasma, or you could integrate it into the chromosome. Okay. You could do either, and then you transform the pool of oligos into your cell, and then you just let it recover. And then you let it recover until it goes to the next exponential growth phase, and then you repeat that step all over again. And so the iteration of that takes about 3 hours in total from one cycle to another cycle. And so this basically eliminates the fact that requirement of having to do selection through a single colony, which has always been a challenge, right? So, you transform the cells and then you plate it out and you have to wait overnight to get it. And so this basically shortens that period, so you can do multiple rounds of this mutation in a very short period of time. And so we thought, well, okay, great. I was doing this by hand every 3 hours, and so there was only so many cycles I could really do in a day. Maybe on a good day I could do 4 or 5. And so it really kind of pushes your physical limits to the max. And so being in the technology-driven lab, George had the really great insight of, well, maybe we should think about actually building an instrument of some sort to try to automate this. And that's where the A in MAGE comes from, so multiplex automated genome engineering. And so I took on the endeavor of trying to actually build an instrument that could do the growing the cells and being able to monitor the growth of those cells using some type of LED photodiode that could measure the turbidity, and then a buffer exchange system where you can automatically prepare the cells for electroporation, which requires washing out all the salts, which is actually a very difficult and challenging task. And then actually doing the electroporation by flowing in oligos and then doing the electroporation, and then taking those cells back into the growth chamber. So that... Though
you built this,
so I had built a number of iterations of this, and so I guess the first it starts out with the first one where you're just kind of trying to use epoxy glue and very kind of garage
do it yourself
bio, and I realized very quickly that the amount of pressure that I was applying to the cuvette was way too high, and I had exploding cuvettes of some sort. So I was like, okay, trying to actually, go through the proper process of design and engineering. And so, for this paper that we had published, we had built something that required a good amount of engineering to get all the details right, to get the temperature control and everything. And so that was quite a learning experience even for me. I didn't think I was going to work on an engineering project where I was building a device.
Influence of your father.
Yeah, that's right. And so, but it was...
Or MacGyver.
That's right. Yeah, I think some combinations of those things, that's definitely true. And so...
Are these now commercially available, these machines?
Yeah, so I think, so we also built a version of that we could actually produce on a larger scale. I think, from a practical perspective, you don't necessarily need a machine to run this protocol. And I think that's one of the kind of the often misconceptions about this paper is that somehow you need an instrument to be able to run this protocol. In fact, you don't. And oftentimes it might not be necessary for you to run tens of thousands of these iterations because the efficiency of incorporation is actually very, very high. And so, we've published a number of kind of protocol papers describing how you could execute the protocol in the absence of an automated machine. So,
right. How do you know when you've achieved the yield you want, how many cycles you have to check periodically by your multiplex PCR? Is that what you do?
Yeah, so that we do that oftentimes just to make sure that we're on the right ballpark.
Okay.
But it's actually quite predictive. The incorporation rates are you can actually model it quite well, and so it starts out being a Poisson distribution, and it kind of becomes a normal distribution very quickly. And so you could that's the nice thing about this, is that you could plan out a large set of experiments where you say, I want to do mutagenesis or engineering of a library of this complexity, and I want to generate this number of mutants, and you can kind of plan that ahead of time and expect to know where you are along the process of doing this, which I think is actually important for mutagenesis studies, where oftentimes maybe you don't really have a good predictive tool, and so you're kind of always flying in the dark about whether you got the mutant that you were looking for or not.
So in this paper, you have a nice example of lycopene.
Yeah.
Well, can you just sort of walk us through that? Tell us what you did. That's pretty neat, I think, unless you think there's a better one.
Yeah, so we wanted to kind of demonstrate this capability, and so one of the things that we thought would be really interesting to show was that you could utilize this technology to introduce mutations that change the translation initiation rates in the pathways that are responsible for making DXP and DMAP, which are kind of intermediates for kind of production of lycopene and other kind of carotenoids. And so we use lycopene also because it's it's found in tomatoes. It's actually what makes tomatoes red. It's an antioxidant, and there's potential properties in terms of its ability for anti-prostate cancer features, and so and also makes the cells red. And so we could see that cells that produce more lycopene turn to a darker shade of red, and which was a great functional selection for us. And so I could actually go in and pick individuals from the population that had an increased production level. So what we did was we combed through the literature and we said, okay, what do we know about the production of these kind of compounds, and what are the genes associated with their biosynthesis or their regulation or the ability for the cells to resist the different types of oxidation states that are associated with increasing toxicity from overproduction of lycopene. And then we accumulated about 30 or so different genes from a variety of different sources. And we said, okay, great. This is what we know about this pathway from the literature. Let's do one experiment where we test all of those in some combination in a parallel path. And so we designed oligos where each oligo was targeting one of such gene, but the oligos would convert the wild type ribosomal binding sequence to a version of the ribosomal binding sequence where we would expect a higher translation rate. And so, but the oligos themselves were actually degenerate. And that's one of the important things about this process, is that you can actually not only introduce one oligo, but you can introduce a pool of oligos. Wow. And that pool, while it's targeting the same location on the chromosome, they actually have different sequences. And you can basically try many, many iterations of this of that location, but potentially in different rates, right? And so we don't necessarily know what the optimal translation initiation rates are for some of these things, because sometimes you might be overexpressing something in a way that would actually lead to a bottleneck in the pathway that actually ultimately decreases the productivity of the pathway. And so we wanted to try all combinations of those things. So we basically had a pool of pools of oligos, right? So we had, each oligo had about 27,000 different variants, and then we had, about 24 of those oligos pooled as one, and then we applied this mutagenesis, and then we got about 3.5 billion variants generated per day, and obviously all of those variants we couldn't quantify, so I plated them out, and then we, from a very subset of those, we were even able to identify, mutants of those that drastically improve the production levels.
And in this case, you have a nice phenotype, red colonies, right?
Exactly. right?
You can tell when it's light red or dark red, right?
Yes.
Up to a certain point, I guess.
Right. So these definitely had higher levels, and that was something that was very interesting. And one of the other interesting things about this is that from the colonies, you could actually see the genome editing in real time, because if you plated the cells at an intermediary stage of the MAGE cycling, you could see the colonies starting from white produce sectored colonies which had different colors, right? And which indicates that individual variants of that single colony was now getting incorporated oligos that then allowed it to have increased production of lycopene. And so that was very gratifying to be able to see that.
So for any process, if a company wants to make a protein in bacteria, they should do this to optimize it, right?
Yeah, so I think this is really useful for when you have some idea of the types of targets that might be relevant for your application. And so, it's not the same as random mutagenesis where you have no idea, right? But this gives you a better anchor on, it's a more hypothesis driven. So I would say it's a semi kind of directed evolution, right? It's not completely directed in that you're still trying combinations of mutations or different configurations of those, but you've drastically whittled your search space to things that are based on what you anticipate might be relevant for the application you're going for.
So how does it do from the perspective of fitness? In the sense, while it's really terrific that you can isolate a mutation, is that mutant then going to be stable in a community where you reintroduce it and, grow it in a 10,000 liter fermenter to make lots of lycopene? Is that mutation sufficiently stable through this process, or do you still have to subject it to further refinement in order to make certain that the clone you first isolated is the clone that you're actually going to spend the money on and put into the 10,000 liter fermenter?
Right. So that's a great question, right? So oftentimes what you develop in the laboratory might not translate into a larger scale when you're doing industrial level productions of any compound. And that's actually one of the big hurdles in the field of kind of fermentation and bioproduction, is that variants that the mutants that you've identified don't scale to a larger kind of size chamber. And this, I think this technique doesn't specifically address this, only because that it's just a very different environment in which you're doing the selection, right? You're not selecting for a large fermenter where the cells need to grow in a particular oxygen level and glucose level. And so that needs to then also be translated to whether these kind of variants are going to be able to perform well in those larger scale reactions. But I think the idea is that if you have the capability to engineer these mutations very quickly, you can then test those different conditions much more easily.
You can scale them.
So it accelerates the rate at which you can test your hypothesis by engineering these mutations. And so, whether those conditions are really relevant in your application still remains to be, tested. And I think that's one of the things that I think it's important to realize about any kind of genome editing or genome engineering kind of endeavors, is that it's really only relevant in the context of a particular environment that you're applying or you're measuring, right? And so, what DuPont uses as a strain to make 1,3-propane-diol on, the million liter scale required a lot of refinement and going back to earlier versions, because they realized that in a larger scale reaction, they needed to have a different genetic background of some sort. But
that's what I was going to ask you. Can you cheat knowing what the background is? Can you bias your selection by the growth medium that you do the original selection on, rather than using something luxuriant like M9 or one of these others? Could you add the fermenter liquor that from a full-scale run as your growth medium, take the liquor out, run it through a filter, and use that to then do your your iteration of the primary mutation.
Yeah, sometimes people do that, right? So, for example, if you're trying to make ethanol, right, for a while, or isobutanol or butanol, right, any of these kind of alcohols, oftentimes there's a lot of toxicity that's built up. So there's a limit to the amount that you can make as a result of the fact that the whatever the cells is making is killing itself, right? And that's going to be a bigger problem when you go into larger scales. And so you can add that as an additional environmental kind of constraint when you're doing the mutagenesis, such that you're simultaneously selecting for individuals who already are kind of are endowed with those more resistant or kind of more beneficial phenotypes. So that's definitely something that people can do. It's not oftentimes that people necessarily think all the way through their entire pipeline to have the vision to be able to do that. But when you do, it certainly could save a lot of time and effort.
That's because we're all still graduate students at heart.
That's right. And there's a lot of biology to be learned. I think that's
I mean, the applications are incredible. You can do so many things. But you did this in E. coli, right? Can you do it in other bacteria, and can you do it in many bacteria at once?
Yeah, so we've also demonstrated doing this in yeast, and so using a very similar protocol, but using a slightly different homologous recombination or kind of a beta protein. And people are now trying to expand this into a variety of other organisms. So one of the challenges is actually that single-stranded oligo transformation is not something thought of as a natural process in the environment. So just not that many people have really tried this type of protocol. But I think there's lots of homologs of the single-stranded binding protein, the lambda beta protein, that we've utilized in natural genomes. And so one may expect that if you transform these oligos into other bacteria, that they could also get incorporated at a reasonably high efficiency. Now,
but you don't need to put in an exogenous single-stranded binding protein?
Potentially. So I think it's all about the level of activity. Yeah. So even in E. coli, there is a background level. So E. coli naturally has single-stranded binding proteins, and so the efficiencies of those were just lower, right? So 10 to the minus 7 as opposed to 10 to the minus 1, which is what we had. And so some of the things that one could potentially think about to do would be to maybe introduce heterologously another single-stranded binding protein that may facilitate this process. One of the interesting things is that actually these beta proteins we don't have crystal structures for them. We only have some EM pictures for these, and so we don't actually know exactly how they're shaping the DNA. We have some kind of good ideas about it, and I think that it remains to be seen whether this is going to be really useful for a lot of other microbes. We think it will be because the protocol in general is quite straightforward. You're just introducing DNA into the cell.
Do you think it will work in mammalian cells?
Yeah, so there was a project actually in George's lab that we had worked on where we tried to do this in mammalian cells, and so it does work. The efficiency was about 1% incorporation, which wasn't bad. The challenge, I think, in mammalian cells in general of introducing single-stranded DNA using this protocol is that mammalian cells see single-stranded DNA as potentially alarming, as in it's a virus, right? Or it's some foreign object that's not that raises alarm. So the cell often go into arrest, right? Growth arrest when they get their incorporation. So what happens is that you get incorporation of these oligos into the genome, but then the cells stop dividing, and the rest of the cells are still dividing, and so your efficiencies are really reduced as a result. But I think those are things that could be overcome with other methods. So,
but is one of the goals one day to do this on, say, gut bacteria taken out of a human or an animal? Is that something that you're thinking about?
Yeah, exactly. I think this is one of a constellation of genome editing tools that have emerged over the last 5 to 10 years. I think we're really at the tip of the iceberg in terms of our capabilities to manipulate.
You mean it's going to get better?
I think it's going to get way better, right?
Yeah.
And I think that's one of the very exciting things about this is that you could, in theory, be able to introduce these oligos maybe not just into one cell, but populations of cells that are maybe different, right? And so you could, instead of doing genome editing on one cell, you do genome editing on a population of cells. And I think that's going to be where a lot of the really interesting biology and the applications are going to be, because oftentimes it's not just a single microbe that's doing something. Sure. It's a community of microbes.
Of course, the problem is in the microbiomes is that not all the microbes grow.
Yes.
Right. So you can't get around that, right?
Right. And I think that's a challenge that I think maybe other ways that genome editing or genome engineering could address. For example, we just actually put out a kind of a opinion piece in Trends in Genetics about this idea of doing in situ genome engineering, which is, instead of domesticating a microbe and bringing it to the lab and doing genetics on it, why don't we come up with a set of methodologies where we could essentially deploy genome editing tools in environments which they're open, right, where the microbes are naturally living, right? And so you could think about it as maybe gene therapy on microbial communities. And I think that's going to be quite important, because it's not going to be often that you're going to be able to recapitulate the growth environments of a complex community in the laboratory setting, even in your gut, right? It works well in your gut, but it's probably not going to work well in a petri dish in the same way.
No, it's going to behave very differently. And if you think about humans not being mice, where everybody eats something a little bit different every day, it's going to be really hard to say, here's a probiotic that we want you to drink that will cure your type 2 diabetes. However, if you could figure out the right gene set or suite in the microbe that is actually responsible for that behavior in the complex community, you could somehow engineer it so that organism that you have, you could treat the human like a fermenter and just drop it in and seeing how it competes in that complex environment.
That's absolutely right. Yeah, I think that's exactly where I think we should be going in terms of thinking about engineering very complex communities where lots of different genes or gene sets are going to be relevant in different combinations. And, in some ways, this is already happening in natural environments, right? So genes move between different organisms through horizontal gene transfer, and they contribute to kind of the increased fitness sometimes, and also into pathological states for things like antibiotic resistance markers that are being shared between different microbes, right?
Or even the dysbiosis that we're seeing with the microbiome in various populations. I was at a seminar last week, and one of the investigators got up and was telling a one-off story about this patient who received a stool transplant from her daughter. Her daughter was pre-metabolic syndrome, and the consequences, the mother had a lean phenotype, and the daughter's stool transplant transformed the mother from a lean phenotype into an obese phenotype.
Oh, no.
Yeah, it took 2 years, and it took 2 years to accomplish this, but the mother was treated for C. diff. Stool transplant worked. She got rid of the C. diff, but the daughter was pre-metabolic. She was in her teenage years. The daughter then went on to develop metabolic syndrome, and the mother then transformed her phenotype from lean to obese. And, there's anecdotes like that all throughout when you talk to your clinical colleagues, but they're ends of one, and no one can explain what the organism was, because the phenotype they were scoring in the clinical event was the treatment successful for alleviating C. diff. And so, I think the risk assessment piece associated with some of these experiments is going to be challenged, as you worry about the unintended consequences.
This episode of TWiM is also sponsored by ASM Microbe 2016. Registration is now open for the all-new meeting, ASM Microbe 2016. You better get your seat before May 5th to take advantage of early bird rates, reduced registration fees. They go up after May 5th. So you do have a little time, but you should get on it before you forget. So now that you hear it, you should go to asm.org/ASMmicrobe, all one word. This meeting brings ASM's general meeting and ICAC both under one roof. These are two separate meetings in previous years. Now they're all together at ASM Microbe 2016. It happens from June 16th through the 20th in Boston, Massachusetts, and it will feature the best microbial sciences in the world and explore the full scope of this wonderful field of microbiology. You'll also have new venues and programs to spark discussion and your interactions with your colleagues in research. And that'll happen all around the meeting campus, which is at the wonderful Boston Convention Center and in the exhibit and poster hall as well. And by the way, we are recording an episode of TWiM and TWiV in front of an audience at ASM Microbe 2016. TWiM is happening in the afternoon on Friday of the meeting time, and Saturday morning around 8 a.m. is TWiV. Get your viruses early in the morning. If your work touches the microbial sciences, this is the meeting you should attend. Learn, connect, and advance at the inaugural ASM Microbe 2016. Again, to learn more and register, asm.org/ASMmicrobe. So, Harris, this idea that Michael said you want to know what genes are important. Sounds like that is something you could tackle, right? The community of genes, of good genes.
Right. Yeah. So we've been thinking about how to do that, and it's oftentimes we have an issue with essentially a lack of understanding of what is really the community is selecting for, right? So we don't know what the actual environment is in your gut, right? Mm-hmm. And so what are the genes that are relevant for enrichment of those traits is not clear.
Right.
We have some hypotheses, things like metabolism of carbohydrates and
right.
fatty acids and things like that. But we don't really have quantitative information about this. And so we've been trying to address some of those with functional metagenomic approaches. So the idea is that if you could heterologously express a library of different gene fragments in a specific microbe, and as a population, and then you can put that into an animal, and then you can see, okay, now that I have the same genetic background of the microbe, but each of them has a potentially unique capability, metabolic or whatnot, that you could look for which genes are being enriched. And if we actually do see enrichment of certain genes that are gut-specific functions. And so things like the ability to utilize different sugars like sucrose, we find that in E. coli, when you put it into the gut, usually E. coli doesn't utilize sucrose, even though it has the transporters to be able to do it. It just doesn't degrade that carbohydrate. Then it has this glycoside hydrolase from a Bacteroides genome that normally utilizes it very frequently. It would actually enhance significantly the fitness of E. coli bacteria in the gut, right? Yeah. And so I think that's the type of information that we really need to get. So we need pools of genes that we can quantify their phenotypic relevance, how important they are in the context of different diets or different genetic background of the host, right, or the different microbiota environments, right? So maybe if you have a dysbiotic gut, it would select for very different genes than if you had a healthy gut, right? And so I think we're really at the beginning of.
So I have this vision of everybody's going to just have E. coli in their gut with lots of other genes from other bacteria. Is that reasonable, or is there something else that having so many different species in your gut gives you besides just the genes?
Well, I think the diversity is an important thing, right? And oftentimes a lack of diversity is linked with a lot of dysbiotic kind of gut diseases. And so we don't necessarily know exactly why it is that the loss of diversity is a signature for pathology, but it somehow is. And I think we need to get at the molecular information and being able to quantify that. But, E. coli is not a major constituent of the natural gut. So, and I don't know how comfortable most people are with having a lot of E. coli in their gut. But there are lots of commensals in the gut that are serving very similar function.
Yeah, because E. coli can get out and then into places it's not supposed to be.
Exactly.
The urinary tract. Maybe another commensal would be better. Right?
Yeah, like Bacteroides for Clostridia, they really don't grow in aerobic environments, so they're not going to go into your bloodstream, for example. Right? So.
Amazing. So, is there anything you could tell us about what's going on in your lab that you'd be comfortable in sharing, just in broad strokes? What kind of? I presume MAGE is still ongoing, and some of these other approaches. What are you doing?
Yeah, so we're trying to extend MAGE also to because you can do mutagenesis very well. Now we can actually start to apply this to look at how the impact of single nucleotide or single residue changes to the fitness of the organism, but you could do that across the entire genome, potentially.
Sure.
But we're doing this one gene at a time, where you could, for example, do saturation mutagenesis across an entire gene by using oligos that target each position. But now with the beauty of next-generation sequencing, you can measure the frequency of those variants within the population, but in very high throughput, right? And by taking different time points under different selective conditions, you can actually now kind of get very quantitative information about the impact of single residue changes on the organismic fitness.
Right.
And I think this is going to be really, really interesting from thinking about how do you design better genomes or kind of better microbes that could function in different contexts. So that's an extension of the types of MAGE applications where we're now thinking about not only can you do mutagenesis, but can you do high throughput quantification of phenotypes, which is going to be the important next kind of complementary set of tools. And so that's something we're pretty excited about.
Did you do that with viruses as well?
Yeah, we haven't done too much with viruses, but that's definitely something that you could imagine doing. So, for example, looking at kind of the viral evolution, of bacteriophages, and how they develop compensatory mutations in the context of even the host mutation, right?
So, right. You have to do it in a context where the virus isn't killing the host, right? Because it has to live long enough to incorporate the oligos, right?
Yeah. So some temperate phages are.
So you're also interested in mice. You have mouse projects. You were just talking about replacing the microbiome with pools of genes, basically, right?
Right. So, we like to think about the mouse as, again, as our petri dish in some ways, especially if you're studying the microbiome or communities of microbes where you really need to have them be growing in some kind of natural, stable environment. And so we're kind of using that as a basis for thinking about how could we introduce genes into those communities. A really nice natural example is that, in Japanese microbiota, so individuals from the Japanese population, they have a specific gene that allows them to degrade porphyrin, which is the kind of major constituent in nori sushi, right? And this gene, which allows them to basically eat sushi, the bacteria to eat sushi, comes from an ocean bacteria that was normally living in the ocean that normally metabolizes seaweed as part of its diet. But that gene was transferred into a gut bacteria some distant time ago. And so this is an example of nature reconfiguring the genome of an endogenous community such that it has enhanced fitness, right? As a result of our dietary kind of changes. And so
another paper we did on TWiM.
Yeah.
That's right.
Yeah.
Exactly.
And so we love to be able to kind of recapitulate this from a synthetic perspective, right? Our diet has changed so much over the last 100 years in the way we process foods and the way that we take medicine and what a lot of things that are being introduced into our gut are very kind of non-natural in some ways, right? And so we need to have kind of a corresponding set of capabilities and tools to be able to interrogate those functions and maybe to reverse it. And I think this type of in situ genome engineering approach is going to be one of a number of applications.
Right. So that seaweed thing is a proof of concept for what you want to do.
It's where I think we're all very inspired by natural processes, and nature is very clever at coming up with solutions that I think we're all trying to chase after in some way.
So one thing that I'm curious about, I mean, you're mentioning tens of thousands of oligonucleotides. When I order an oligo, it's $1 a base, and that's a lot of money. We must get a deal or something, right?
Yeah, well, so nowadays that's actually a very interesting point, right? Because just as DNA sequencing has really reduced in cost, DNA synthesis has also been reducing in cost, right? And so it's a lot cheaper today to get an oligo than it is, even 5-10 years ago. I mean, when I was George Church has a really great slide about this, where back in 1980. They had ordered an oligo that was a 15 base pair oligo to the Harvard labs, and it costed something like $3,000. And
I bought some of those.
Yeah, and it required 6 to 8 weeks of lead ship time just to get that, right? And that's orders and orders of magnitude cheaper now than it is just even maybe a decade ago. And I think we're getting even better at it now with a lot of these oligos that are being synthesized off of oligonucleotide microarrays, right? So these chip-based synthesis approaches are really driving the cost down, and so it's really about cost and scale, right? And so if there's more demand for synthesis-like applications, the cost is going to, it's the economics really work in that in your favor there. And so, with all of these synthetic biology approaches of trying to design your own genome and synthesize your own genome and testing lots of variants, I think there's a growing market for cheaper oligos, which really helps with this. So
it's not the biggest component of your budget, is what you're saying.
It's not the biggest component, although it's still a big component of the budget. But
it probably is per diem.
That's right. That's right. It's certainly something that I think the community, over time will resolve by volume. So,
when I was a graduate student in the 70s, oligos were just starting to be synthesized. They were very expensive, as you pointed out. And I remember my advisor saying, he said, one day there will be an insatiable demand for oligonucleotides.
That's probably true, and to some extent. I think I have a line in my thesis. When I was putting it together, I said something like, one day DNA and nucleotides will be traded like a commodity that you would like wheat or with gold, and because if you think about kind of the revolution of the green technologies and all of these things, it could potentially be a commodity of some sort, right? But obviously that's highly speculative. But
well, you're saying that one day you're going to know which oligos you should eat to transform the bacteria in your gut to do good things? Is that a feasible
I think
idea?
that's what he's saying. I think that's what he's saying. He's going all the way in.
That's right. Yeah. So I think we're gonna get there. But we really don't know how deep the hole goes, and and so then that's a kind of the exciting part of doing this work, is that we might just be at the tip of the iceberg.
Of course, it sounds like it, absolutely. I wonder if I could ask you one last thing, which is a little bit different, but I think our listeners would love to hear this. This is your PLOS One paper that was covered in the Wall Street Journal. It's called An Economic Framework of Microbial Trade. Can you explain that to us?
Yeah, sure. Yeah, this is one of the really fun projects that I worked on, and this is in collaboration with Josh Tassoff, who's an assistant professor in Claremont Graduate University in California. And we were actually
he's an economics professor.
He's an economics professor. He's a full-blown economist, and so the origin of this work actually comes from that we were actually both at MIT and we were roommates, and so so this is
that's how we got there.
That's okay. Right. So we had talked about it would be really interesting to think about the interface between these two fields, and it was funny because I think Josh had he wanted to study biology and evolution as an undergraduate at MIT, but then he realized all the quantitative approaches for studying that were all in economics for some reason, and so he became an economist. But he always wanted to go back and study biology, and so at some point we met up in New York, and it was and we started talking more about our work, and we realized there was a large parallel between economic principles and the way we think about trade of goods and services, and the way that microbes exchange metabolites and resources, and what are the drivers of those exchanges. And so we decided to start thinking about this and actually developing a model for describing how microbes exchange metabolites and resources in a very economic framework. And so it's kind of a fusion of two disciplines. And I think, again, it's one of these things where I think it's, there's lots and lots of well-established principles in economic theory, right, that we could potentially apply in terms of describing microbial communities. And, I didn't appreciate this also from an economist perspective. Biology is a very interesting system if the economic principles do apply, because it allows you to now test some of the hypotheses that economists have generated that you can't really test in a real setting, because you can't change world markets in the same way that you can change.
So we could actually do Ronald Reagan's trickle down theory. We could actually test trickle down theory.
Yeah, some version of that one could start to think about that. But
that would be an interesting experiment, Mike.
It would be. This is Vincent gave us a batch of papers to look at, and they were all fascinating. But this one found a special place in my heart, and I'm going to actually torture my poor graduate students in the department and actually have them do a journal club on this one to revisit some of the math and to revisit some of the discussions, because I think everybody has a sense of what they think economics is, and really putting the biology into something that people have intuition about will help them in their own science as they begin to think about their experiments and other things. So if you, and since this is in PLOS One, it's open access, so everyone can see this paper and I really commend this to the listeners, because it's a really interesting read if you're doing microbial physiology or ecology or even economics. And I just found it absolutely fascinating. And I thank you for writing it.
Oh, thank you. Yeah, it was a really joy to put it together, because you really have to think about what are the important kind of ideas and how do you translate them between two very different fields. And we spent a lot of time thinking about that and I was trying to understand what Josh was saying, and he was trying to understand what I was saying, and even just the jargon, that's the academic jargon that's utilized is not necessarily the same. And so you kind of have to kind of break those barriers.
There's even an experiment in this paper with bacteria, right?
Yeah, we did an experiment too. So
measurements of growth, abundance, trade-offs. The idea being that countries make things and trade them, right? And that controls abundance of materials, their price, and so forth. Bacteria do the same thing, right?
Yeah. So the idea is that you can be altruistic and share your resources by trade, right? And the idea is that then you could potentially get a reciprocal benefit if the partner that you're trading with is able to produce a metabolite that you're not able to produce, or you're not able to produce well. And so, but there's some type of trade-off between how much you should share, right? And if you share too much and be very generous, it's to your detriment.
Sure.
And so there's some kind of equilibrium. So one needs to reach. And so obviously, if you don't share at all, then it doesn't benefit anyone, right? But if you share too much, then it also doesn't benefit everyone well. And so, there's that kind of optimality is what we were trying to kind of explore. And it's actually quite interesting, because it's this idea that if I trade with you, I might gain a little bit of benefit, but you might actually get a lot more benefit. But even in those circumstances, it's still better for me to trade, because if I didn't trade, I would be worse off. Right. And so there's this kind of interesting trade-off of, well, should I, how much should I be benefiting those around me, given that they might even grow more than I will. Right. But if I don't grow at all, it doesn't help me. So, it's almost like this forced cooperation. So
I think you've answered your own question as to why diversity in our gut is important, because this paper goes to support that contention.
Yeah. So, I mean, it's
all about trade, and microbes trade goods and services amongst themselves in our robust gut of 1,000 to 5,000 unique individuals that are exchanging or doing commerce. And if you think about the globe having between 130 and 150 true economies, and if you throw California in there, the global economy is the equivalent of the mammalian gut. I wonder if the Wall Street Journal got that.
I don't know. I couldn't read the article. It was behind a paywall.
Oh. Even at Columbia, you can't get Wall Street Journal?
I can get all the science journals here, but not the Wall Street Journal.
Yeah.
Harris, how does this apply to bacteria, endosymbiotic bacteria that have to live inside another? They share metabolic pathways. Are there economic equivalents of that?
I think there is. Yeah. It's going to be much more of a syntrophic interaction, right? Where they have to be intimately interconnected in their trade. But that's certainly true in many nations or firms where their businesses are completely interdependent on other agencies, right? And so it's like you can't make an iPhone without Foxconn production, right? Or this particular lens from somewhere. And so I think that's actually a really interesting kind of opportunity to think about, and things like vertical integration versus kind of horizontal, being a jack of all trades versus specializing in a specific sub area, and how microbes choose to do that, or why they're selected to do that as opposed to be generalists. I think it's really fascinating, and I think this is really the kind of the beginning of a long list of things that we hope to be able to try to tackle. So
I think it's interesting because both fields will probably benefit from this, both microbiology and economics, right? Because you get a different kind of input for both.
Yeah, that's right. That's right.
Have you gotten people emailing you and interested in this, and do you think it sparked something out there?
Well, yeah, I mean, I think certainly so. I mean, the fact that the Wall Street Journal, was interested in writing about it, I think shows that, economists and people in the financial sectors and just in general like to think about the parallels of these principles in other communities and other areas. And so we've gotten kind of really interesting kind of feedback from people, and it's it's an emerging kind of topic. I think there's going to be lots of different ways to think about and tackle this problem, and so I'm pretty excited for this. And I was very fortuitous, I guess, that my roommate in college happens to be an economist, so we kind of were able to sit down and actually put together an interesting piece.
It goes to my old adage: life is cumulative.
I'm just loading the paper here to try and see the metrics. We'll see how many people have downloaded this, but it's not coming
Plus
up.
one's not cooperating.
Yeah, the metrics are spinning, I don't know why that would be. It's loaded the paper already. They should load the metrics. And they have to pay for their servers. It's not cheap. Gentlemen, any other questions for Harris before we sign off here? You guys have
I learned too much today.
You learned too much? Can't learn too much, but I certainly learned a lot. I hope. And thank you, Dennis, our listener, Dennis. Thank you for sending us that question that got us to this point. And I'm sure, Harris, we'll have you back sometime when you publish some more papers from your laboratory with even more specific results. It'll be cool. And this episode of TWiM, like all the others, you can find at iTunes, microbeworld.org, and also microbe.tv/TWiM. Please send us your questions and comments, but not about economic theory. If you do, I'll send them to Harris. You can send them to TWiM at microbe.tv. I want to thank our participants today. Elio Schaechter, you can find him at his blog, Small Things Considered. Thank you, Elio.
It was enlightening today.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you.
And I'm jealous of your weather, but we'll get there.
You'll get there soon.
At some point. Harris Wang is right here at Columbia University Medical Center. Thank you, Harris.
Absolutely. Thanks so much. It's great to be back in this office.
I hope you don't leave after 6 more years.
No, we hope to be staying here.
But you're going to move every 9 years, you said. Or is that it for now?
Well, my trend broke when I stayed in Boston for an extra couple of years. So I think I no longer have to keep that trend there.
Yeah, I wish I could see you more. There's no good reason because you're only a 10-minute walk away, but maybe...
We'll definitely have to make it a more regular
Try
thing.
and figure out a way to do that because I enjoy our conversations. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM, and in particular Chris Condayan and Ray Ortega for their technical help. The music you hear on TWiM is composed and performed by Ronald Jenkins. Check out his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.